This is a Fauzaan's IB video on DNA replication. The IB themselves have a couple of understandings when it comes to DNA replication. The first being DNA replication is continuous on the leading strand and discontinuous on the lagging strand, as well as DNA replication is carried out by a complex system of enzymes. So the two terms leading strand and lagging strand we will attempt to explain over the course of this video, and also over the course of this video our objectives are to understand what the role of the following enzymes in DNA replication, as well as understand how free-floating nucleotide, oxidized fragments, and RNA primers, how do they relate to DNA replication. So to explain DNA replication, we can have a look at this diagram. It may look complicated at first, but we can depict it and consequently understand how DNA replication works. So on the right, we can observe a parental DNA helix entering what is known as a replication fork. Replication fork, excuse my mouse writing. And then after going through this entire process in the middle, we end up with two pieces of double-stranded DNA at the end. Thus, DNA has been replicated. Now, before we can truly understand how DNA replication works, there are a couple of pieces of background knowledge that we need to understand. The first being that DNA replication is semi-conservative. What this essentially means is that part of the parental DNA can be found in the final product, as opposed to it being the final product being entirely just replicas. So this is apparent here because if we notice, the parental DNA is green, and at the end of this, the strand is half of the old green and half the brand new red. And over here, the other strand is also the same thing. Half of it is the old green, and half of it is the brand new red. The second thing to understand before we begin is the idea that DNA is anti-parallel. Now this is apparent in the diagram on the right that we've got here. If we have a look at this blue strand, the blue strand from the bottom to the top of the page is going from 3 to 5. Whereas the yellow strand is going from 5 to 3. And both of these strands are held together by hydrogen bonds. Hydrogen bonds tend to be very strong, and if they are broken, under normal circumstances, they will attempt to bind back to each other. This premise is also very important in DNA replication. Another thing to understand is that DNA is tightly wound in a double helix, which can be observed in this diagram here. Now, the issue with this is that it makes it significantly more difficult to create this entire process. So the job of the first enzyme in the process, DNA gyrase, it works to relieve tension in this double helix. It's essentially making it more relaxed, so to speak, and allowing the rest of the enzymes to work. Now, this, like we said, this diagram can appear very complicated, but we can break it down into sections and we can understand what is going on. If we first take a look at this section here, the first section that goes on, where the parental DNA helix, helicase, and the single-strand binding proteins, the job of helicase itself is to break the hydrogen bonds within the double-stranded DNA. So as you can see here, the parental DNA first begins double-stranded, and then it's separated into two separate strands here. However, we established earlier that under normal circumstances, as hydrogen bonding is very strong, those bonds will just form back again. However, we are under the presence of single-strand binding proteins. I can find my mouse. Single-strand binding proteins. This doesn't occur. These enzymes are responsible for making sure that these strands do not bind bind back together, but instead the strands bind to the enzymes. These enzymes hold the strands apart so that they cannot bind back together with hydrogen bonding. So now at this point we have we've taken the parental DNA helix, we've put it through the replication fork, and we have two pieces of single-stranded DNA. This is no good to us as we want two pieces of double-stranded DNA. The essential premise of DNA replication and the essential enzyme that conducts DNA replication is DNA polymerase 3. Now, DNA polymerase 3, the essential job of DNA polymerase 3 is it'll read the parental DNA, it'll recruit free floating nucleotides from the area, and then it will bind them onto the parental DNA, turning a single-stranded piece of DNA into double-stranded. Now, something that might cause us an issue, and which we can explain, is the idea that DNA polymerase 3 reads in a 3 to 5 direction, and consequently builds in a 5 to 3 direction, as DNA is anti-parallel. On the leading strand up here, we can see that next to the 3 we have next to the green strand we have a 3, and DNA polymerase 3 reads in a 3 to 5 direction. So it's not a problem as more DNA comes through the replication fork towards DNA polymerase 3. It'll just read back towards the replication fork and consequently just keep on working. The strand is being built continuously. However, the issue lies on the lagging strand. As DNA strands are anti-parallel, we had the 3 up here, so we have the 5 down here. As a result, DNA polymerase 3 can only run in this direction, as we have a 3 up here next to the replication fork, and we have a 5 down here at the end of the DNA strand. So this becomes an issue, as DNA polymerase 3 can only build away from the replication fork. However, new single-stranded DNA is constantly coming through. So the way that we overcome this problem, if we just have a look at the bottom half of this diagram, is via a lot of different enzymes. We use DNA primase, DNA polymerase 1, and DNA ligase. DNA polymerase 3 needs a small area of double-stranded DNA to bind to the single strand. From there, it will just keep building, but it needs an area of double-stranded DNA to bind. Now, what primase does, primase attaches an RNA primer to the single-stranded DNA, making it double-stranded in a small area. Now, this allows DNA polymerase 3 to bind and consequently start building away from the replication fork. Now, obviously the problem is we don't want RNA primers, we want DNA, we want double-stranded DNA. For that, we use the enzyme DNA polymerase 1. DNA polymerase 1, its job, or what it does, is it replaces the RNA primer with a DNA primer. Now, we still haven't finished here, because we are constantly adding primers, building away from the replication fork, adding primers, building away from the replication fork, replacing the primers. But all these discontinuous fragments have to be joined back together. These discontinuous fragments are known as Okazaki fragments, and they are joined back together by DNA ligase. DNA ligase is responsible for joining these Okazaki fragments back together. So to once again go over it, on this lagging strand, as single-stranded DNA passes through the replication fork, primase attaches an RNA primer. DNA polymerase 3 can therefore attach to the single-stranded piece of DNA and recruit free floating nucleotides from the area and use the parental strand as a template and then build on it. Following that, the RNA primers are replaced with DNA primers by DNA polymerase 1. And then these Okazaki fragments are now joined together by DNA ligase. And then from the entire process, we end up with 2 strands of double-stranded DNA replicated semi-conservatively. Now, this entire process occurs in multiple different locations along a strand of DNA. So, and what we refer to these as are replication bubbles. So it will occur here, it will occur here, it will occur here on this double-stranded DNA. And the reason this occurs is just to make the entire process more efficient. So instead of just replicating one, this diagram is replicating in 3 different areas at once. So therefore, it'll get the entire process done 3 times as quickly. These replication bubbles expand in both directions, which eventually will leave you with two daughter DNA molecules. So that is the entire process of DNA replication. Okay, to summarize, DNA gyrase works to relieve tension in the DNA helix so that helicase can separate the 2 strands by breaking hydrogen bonds. To keep these 2 strands separate, single-strand binding proteins are used, as if they weren't kept separate, the hydrogen bonds would just reform. DNA polymerase III catalyzes the formation of a new strand. It uses each parent strand as a template and adds complementary bases to form new strands as it moves along the parent strand. The 314 nucleotides are available to be recruited. DNA polymerase III only moves in one direction. It moves in the 3 to 5 direction and consequently attaches nucleotides in the 5 to 3 direction. And due to the fact that DNA strands run anti-parallel, the DNA polymerase III works in different directions on both strands. On the leading strand, it moves towards the replication fork and builds the new strand continuously. However, on the lagging strand, DNA polymerase III moves away from the replication fork and consequently the strand is built discontinuously in small sections known as Okazaki fragments. A primase is used to attach a primer to the single strand. It's a small region where single strand becomes double-stranded, and DNA primase achieves this by using RNA nucleotides, not DNA nucleotides, RNA nucleotides, and consequently they have to be removed later. These small double-stranded areas allow DNA polymerase III to bind. This only has to occur once on the leading strand. However, it has to occur multiple times on the lagging strand, as on the leading strand it's continuously built, whereas on the lagging strand DNA polymerase III has to unbind and keep rebinding constantly. DNA polymerase I removes the RNA primers from the lagging strand and replaces them with DNA nucleotides. So now we just have Okazaki fragments of DNA nucleotides, and DNA ligase works to join the Okazaki fragments together. So at this point, we have 2 strands of double-stranded DNA. Now this occurs, these replication forks, there are multiple of them present along a single DNA molecule to make this process more efficient. And these replication bubbles occur in both directions, so eventually you will be left with 2 strands of double-stranded DNA. And that is DNA replication. Thank you for watching.